Have you ever marveled at your body's, incredible ability to store vast amounts of energy far more efficiently than any battery we've ever invented?
It's pretty amazing.
Or wondered how it manages to tap into those reserves when you haven't eaten for hours, keeping your brain sharp and your muscles moving without missing a beat? It's truly an engineering marvel.
It really is.
Today, we're taking a deep dive into the fascinating world of fatty acid, triacylglycerol, and ketone body metabolism.
Mm.
We're not just going to cover how your body makes and breaks down fats. We're going to explore why these processes are so vital for everything, from constructing the very foundation of your cells to powering your brain during a prolonged fast. Get ready for an exploration that will hopefully make you truly understand the mastery behind your own internal energy systems.
Absolutely. Our mission for you today is to extract the most crucial interconnected insights from this complex topic. We've compiled a comprehensive review of the biochemical pathways, allowing us to go beyond just the basics. We want you to walk away with a profound, almost professional level understanding, appreciating the intricate dance of these molecules and enzymes, not just what they are, but how they collaborate. We'll uncover the surprising roles these molecules play, and perhaps more importantly, what happens when this delicate dance gets, thrown off balance.
Yeah, because it's not just about memorizing facts, is it? It's about grasping the elegance and resilience of your own biology. We'll demystify what can often seem like dense biochemistry and transform it into, hopefully, a captivating story of energy, structure, and survival within your own cells. Let's unravel the secrets together.
Sounds good.
Okay, let's start by introducing our main characters: fatty acids and triacylglycerols. These are truly fundamental to how your body operates. Fatty acids, the building blocks, primarily exist in two forms in your body.
Yeah.
Either free-floating, what we call non-sterified, or incorporated into more complex molecules as fatty acyl esters. They are surprisingly dynamic, constantly moving around.
That's absolutely right. These free fatty acids, or FFAs as they're often called, are found in small amounts pretty much everywhere in your tissues. But during times of fasting, their levels in your blood plasma can really ramp up quite significantly.
Oh.
Think of them as nimble, swift energy couriers, always on the go. They're not just floating freely, though. They're transported through your bloodstream, securely attached to a protein called serum albumin.
Albumin, right.
Yeah, which acts like their dedicated transport vehicle. Now, where do they come from? They're either released from your main fat stores, the triacylglycerols or tags in your adipose tissue, or they come from other circulating lipoproteins. And where are they headed? Primarily to tissues that need energy, like your liver and muscles, where they're burned for fuel. Your liver also has a special role. It takes a significant portion of these FFAs and converts them into ketone bodies.
Ketone bodies, we'll get to those later, I assume.
Exactly. They become a crucial alternative fuel, especially for your brain during longer fasts. We'll definitely dive deep into ketones later on.
So they're not just fuel delivery trucks, but incredibly versatile ones, constantly being dispatched where needed. But what's truly fascinating is that their role extends far beyond simply providing energy, doesn't it?
Absolutely. While their energy-providing function is immense, fatty acids are also fundamental architects within your cells. They form the backbone of membrane lipids, like phospholipids and glycolipids, which are, the primary components of all your cell membranes.
The actual structure of the cell wall.
Precisely. Without the right fatty acids, your cell's outer layers simply wouldn't hold together or function properly. They also play a critical role in enhancing how certain proteins interact with these membranes, which is vital for countless cellular processes. And here's another surprising role. Fatty acids are precursors for a whole family of powerful hormone-like signaling molecules called prostaglandins.
Prostaglandins, right. Involved in inflammation and things like that.
Exactly. They're involved in a vast array of bodily functions, from regulating inflammation and blood clotting to influencing blood pressure. It's really a testament to their versatile design.
So beyond just fueling your body, they're like the unsung heroes of your cell's architecture, ensuring everything holds together and communicates properly. That's a level of importance many might not realize.
Precisely. And speaking of storage, that brings us neatly to triacylglycerols, or tags. These are essentially 3 fatty acids neatly packed onto a glycerol molecule. They are predominantly stored in your white adipose tissue, basically, your body fat. And they represent the largest and most efficient energy reserve your body possesses, bar none. When you look at the broader landscape of metabolic health, alterations in how your body handles fatty acid metabolism and tag storage are directly linked to some of the most pressing health challenges of our time, like obesity and type 2 diabetes. Understanding these pathways isn't just academic, it's really key to understanding well-being.
That's a powerful connection. We're talking about the fundamental ways your body manages its energy currency.
Yeah.
Grasping these pathways is absolutely critical for understanding health and disease and how diet and lifestyle really impact what goes on inside our cells.
Indeed. It's like seeing the grand master plan, you know? If you could visualize the entire metabolic network, you'd see how the synthesis and breakdown of tags are intricately connected to carbohydrate metabolism. Everything is interwoven, creating a dynamic, responsive energy system within you.
Okay, let's zoom in on the structure of these fatty acids because, as with so many things in biology, the tiny details here have huge implications for their function. So, if we were to draw a fatty acid, what would it look like?
Well, at its most basic, a fatty acid is composed of two distinct parts. Imagine a long chain of carbons, usually quite long, that's hydrophobic, meaning it really tries to avoid water.
The tail.
Exactly, the tail. At one end of this tail, there's a small special group called a carboxyl group. This carboxyl group is ionizable, meaning at the neutral pH inside your body, it typically loses a proton and becomes negatively charged, COO. This charged end is hydrophilic, or water-loving.
So it's like a long, water-fearing tail with a tiny, water-loving head, giving it a sort of split personality.
That's an excellent way to put it. Because of this dual nature, fatty acids are called amphipathic. However, for the longer fatty acids, which are most common, that water-fearing tail is so much larger than the small water-loving head that these molecules are highly insoluble in water overall.
Right, they can't just float around freely.
Exactly. They can't just float freely in your blood plasma or they'd clump up and cause all sorts of problems. That's why they need transporters. In fact, over 90% of the fatty acids in your plasma are actually in the form of fatty acid esters like tags, cholesterol esters, and phospholipids, all neatly packaged within lipoprotein particles.
Like little delivery trucks.
Precisely. Sophisticated molecular delivery vehicles. The free fatty acids, those without the ester linkage, are transported by binding to albumin, your most abundant serum protein, a real workhorse of the bloodstream.
That perfectly explains how these fat-based molecules navigate a watery environment like your blood without causing issues. Now, let's talk about their internal structure. Are these carbon chains perfectly straight, or do they have bends, and why does that matter?
This brings us to the concept of saturation, which is absolutely critical. Fatty acid chains are saturated if they contain no double bonds between their carbon atoms. They're fully loaded, you could say, with hydrogen atoms.
So they're straight.
Like straight, rigid rods, essentially. But if they contain one or more double bonds between carbons, they are unsaturated. One double bond makes them monounsaturated. Multiple double bonds make them polyunsaturated. Now, in humans, the vast majority of naturally occurring double bonds in fatty acids are in the cis configuration rather than trans.
Cis. Okay, what does that mean structurally?
And here's where the magic happens. A cis double bond introduces a distinct bend, or kink, into the otherwise straight hydrocarbon chain. If a fatty acid has multiple double bonds, they are typically spaced precisely at 3 carbon intervals, and each one introduces another kink.
So it makes the chain less straight, more bent.
Exactly. This seemingly small structural detail has profound implications for how fatty acids behave. Critically, these double bonds decrease the melting temperature of a fatty acid, making it more fluid at body temperature. Conversely, increasing the chain length generally increases the melting temperature. So, because your cell membranes are made up of many long-chain fatty acids, the presence of these cis double bonds and their kinks helps maintain the vital fluidity of those membrane lipids.
Fluidity is key for membranes, isn't it?
Absolutely essential. Imagine your cell membrane as a dynamic, flexible structure rather than a rigid, brittle wall. That flexibility is crucial for its function, allowing proteins to move and interact properly within the membrane.
Here's where it truly gets insightful. These tiny kinks are absolutely vital for how flexible and fluid your cell membranes are. It's like the difference between building a wall with perfectly straight bricks versus slightly angled ones that allow for more movement and adaptability. That bendiness is crucial for cell function, from nutrient transport to cell signaling.
Exactly, and this structural diversity leads to precise naming conventions. Most fatty acids in humans have an even number of carbon atoms, commonly 16, 18, or 20, though it's worth noting that your brain has a special capacity to produce very long-chain fatty acids, over 22 carbons, which are critical for its unique lipid structures.
VLCFAs, right?
Correct. When naming them, we start numbering carbon atoms from the carboxyl end. with the carboxyl carbon itself being carbon 1. So if you see something like 20.4, 5,080, 1114 for arachidonic acid, the 20 means it has 20 carbons. The 4 means 4 double bonds.
Okay.
And the numbers in parentheses 5, 8, 11, 14 tell you exactly where those double bonds are located relative to that carboxyl N. Carbon 5, 8, 11, and 14.
It's like a sophisticated GPS coordinate system for each fatty acid, telling you its size and where its kinks are. Very precise.
It truly is. We also use older but still very relevant Greek letter designations. Carbon 2 is the alpha carbon, carbon 3 is the beta carbon, and so on. But a particularly crucial one for nutrition and metabolism is the omega carbon. This is the carbon of the terminal methyl group at the very end of the chain, regardless of how long the chain is.
The tail end.
The absolute tail end. This gives us the omega designation, or sometimes the N designation, for double bond position. For example, when you hear about omega-3s or omega-6s, you're referring to where the first double bond is located, counting from that omega-N.
From the methyl end, not the carboxyl end.
Exactly. An omega-6 fatty acid has its first double bond 6 carbons away from the omega end, and omega-3 has 3 carbons away. You might also see this as N6 or N3, which means the same thing.
So all those discussions about omega-3s and omega-6s in your diet are really about precisely where that last double bond is located from the very end of the fatty acid chain. That brings a whole new level of understanding to nutrition labels for many people.
It absolutely does. And this concept of double bond position brings us directly to a critically important topic: essential fatty acids. Linoleic acid, which is an omega-6, and alpha-linolenic acid and omega-3 are considered dietary essentials for humans.
Meaning we can't make them ourselves.
Exactly. The reason is profound. Your body simply lacks the specific enzymes, the desaturase, that can introduce double bonds beyond carbon 9, specifically from carbon 10, all the way to the omega end.
So we have a limit on where we could put those kinks.
We do. This means we cannot synthesize these fundamental molecules from scratch. We are entirely dependent on getting them from our diet, primarily from plants. Interestingly, arachidonic acid, that important omega-6 fatty acid we mentioned as a precursor for prostaglandins, only becomes essential in your diet if you're not getting enough linoleic acid.
Because we can make it from linoleic.
Correct. Your body can make arachidonic acid from linoleic acid, provided you consume enough linoleic acid. Alpha-linolenic acid, the omega-3, is a crucial precursor for other important omega-3s involved in growth and development. Now, while essential fatty acid deficiency is rare in industrialized nations with varied diets, it can lead to serious health issues if diet is severely restricted. The most notable symptom is a dry, scaly dermatitis. This is because these essential fatty acids are crucial for synthesizing specific molecules, ceramides, that form the water barrier in your skin, keeping it hydrated and protected.
So your diet isn't just about calories or macronutrients. It's literally about getting these absolutely critical building blocks that your body cannot make on its own. It's a powerful reminder of how finely tuned our nutritional needs truly are, impacting everything from your brain to your skin.
Well said.
All right, now that we understand what fatty acids are and why some are essential, let's pivot to how your body actually builds them from scratch, a process we call de novo synthesis. What's the big metabolic strategy behind this, and where does it happen?
Right. The why behind fatty acid synthesis is really a brilliant energy-saving strategy. When you consume more carbohydrates and proteins than your body needs for immediate energy or structural repair, your body cleverly converts that excess into fatty acids. These fatty acids are then stored as tags, providing a much more compact and efficient long-term energy reserve compared to, say, carbohydrates like glycogen.
So it's like packing away extra fuel for a rainy day.
Exactly, preparing for potential future energy needs. As for where, this process primarily takes place in your liver, which is a real metabolic powerhouse. It's also very active in lactating mammary glands, obviously for synthesizing fats for milk production. And adipose tissue, while primarily a storage site, also has some capacity for synthesis, though generally less than the liver in humans.
And where in the cell does this happen?
The entire process occurs in the cytosol, the fluid portion of your cell, outside the mitochondria. It requires an input of energy, making it endergonic, and it's a reductive process, meaning it requires electron donors, specifically NADPH, to build up these long carbon chains.
So it's your body's clever way of converting excess dietary fuel into a highly condensed long-term storage form, like preparing for a long journey, even if you're just sitting on the couch after a big meal. It's smart energy management.
It absolutely is. But there's an initial hurdle, a logistical challenge, you might say. The primary carbon source for fatty acid synthesis is acetyl-CoA.
which comes from breaking down glucose or amino acids.
Correct. And the acetyl-CoA is mostly generated inside your mitochondria. However, the CoA part of acetyl-CoA can't cross the intermitochondrial membrane to get to the cytosol where fatty acid synthesis actually occurs.
So how does the useful acetyl part get out?
It uses what's called the citrate shuttle. Acetyl-CoA combines with oxaloacetate, or OAA, inside the mitochondria to form citrate, the first step of the TCA cycle. Crucially, citrate can be transported across the mitochondrial membrane into the cytosol via a specific transporter. And this citrate shuttle itself is a beautiful piece of cellular communication. Citrate is transported out of the mitochondria into the cytosol, specifically when mitochondrial citrate concentrations are high.
And when would that happen?
That happens when your cells have ample ATP, signaling a high-energy state. High ATP levels actually inhibit a key enzyme in the TCA cycle, isocitrate dehydrogenase, causing citrate and isocitrate to build up.
So the buildup itself is a signal.
Precisely. The presence of citrate spilling into the cytosol acts as a powerful high-energy signal, telling the cell, we've got plenty of energy, let's store some as fat. Once in the cytosol, citrate is then broken back down into OAA and acetyl-CoA by an enzyme called ATP citrate lyase. This requires ATP, but it effectively makes the acetyl-CoA readily available for fatty acid synthesis right where it needs to be.
That's a truly elegant control system. High energy levels trigger the body to actively store that energy as fat. It's a sophisticated, built-in energy management system that knows when to save. Very clever.
It truly is. Okay, so once we have acetyl-CoA in the cytosol, the very first committed and most critical step in fatty acid synthesis is its conversion to malonyl-CoA. This reaction is catalyzed by acetyl-CoA carboxylase, or ACC. This enzyme is incredibly important because it's the rate-limiting step and the primary regulated point in the entire pathway.
The main control knob, essentially.
You got it. This reaction requires bicarbonate as the source of the added carbon, ATP for energy, and the vitamin biotin, also known as vitamin B7, as a crucial coenzyme that carries the activated CO2.
So this ACC enzyme is like the gatekeeper of fat synthesis. It dictates how fast your body can build new fatty acids, acting as a crucial control point.
Exactly, and because it's so critical, ACC is under very tight short-term and long-term regulation. In the short term, it's allosterically activated by citrate.
The same citrate that carried the acetyl-CoA out.
The very same. Citrate binding causes inactive ACC enzyme units, called protomers, to polymerize into long, active filaments, really revving up synthesis. Conversely, it's allosterically inhibited by palmitoyl-CoA, which is the typical end product of fatty acid synthesis.
Feedback inhibition.
Classic feedback inhibition. High levels of the end product signal to the enzyme, we have enough, slow down, causing the active filaments to depolymerize back into inactive protomers. There's also crucial covalent regulation through phosphorylation, an enzyme called adenosine monophosphate activated protein kinase, or AMPK, phosphorylates and inactivates ACC.
AMPK, that sounds like an energy sensor.
It is. AMPK itself gets activated when cellular energy levels are low, specifically when AMP levels rise relative to ATP. It can also be activated by other kinases, like protein kinase A, PKA, which is dependent on cyclic AMP or AMP. This links ACC regulation directly to hormonal signals. For instance, when counter-regulatory hormones like epinephrine, your fight-or-flight hormone, or glucagon, released during low blood sugar are present, they raise KMP levels, activating PKA. PKA can then activate AMPK, which in turn phosphorylates and inactivates ACC, effectively putting the brakes on fatty acid synthesis. You don't want to be storing fat when you need to mobilize energy, right?
Makes perfect sense.
In stark contrast, when insulin levels are high, typically after a meal signaling energy abundance, ACC is dephosphorylated and activated, strongly promoting fat synthesis to store that excess energy.
So when your body is low on energy or needs a quick fuel boost, it literally puts a stop to fat storage. And when insulin is high after a big meal, it signals store, store, store. It's a brilliant push-pull system ensuring energy is managed perfectly according to supply and demand.
It really is a masterpiece of reciprocal control. And beyond these immediate short-term controls, ACC also undergoes long-term regulation through changes in its gene expression. If you consistently eat a diet that's high in calories, particularly one rich in carbohydrates and low in fat, your body will actually increase the synthesis of the ACC enzyme itself, boosting its overall capacity to make fatty acids.
Adapting to the long-term dietary pattern.
Exactly. Conversely, prolonged low-calorie diets or high-fat, low-carbohydrate diets will reduce ACC synthesis. This long-term regulation is influenced by specific transcription factors, proteins that control gene activity. Key players here are ChREBP, which senses carbohydrate and glucose levels, and SREBP1C, which is strongly induced by insulin. It's worth noting that the enzyme responsible for the rest of the synthesis pathway, fatty acid synthase, or FAS, is regulated similarly at the gene level by these same factors.
So the whole production line gets ramped up or down together.
Yeah, precisely. And this has profound clinical connections. Consider metformin, a very common medication for type 2 diabetes. One important way it helps lower circulating triacylglycerols in the blood is by activating AMPK. This not only inhibits ACC activity directly through phosphorylation, but it also decreases the overall expression of both ACC and fatty acid synthase genes, partly by reducing SREBP1C activity. Plus, metformin also helps lower blood glucose by increasing glucose uptake by muscle cells, also mediated by AMPK.
This truly illustrates how deeply diet, energy balance, and even medications can influence your body's entire fat-making machinery. It's a complex and finely tuned interplay, highlighting that every choice we make impacts these systems. Very interconnected.
Absolutely. Now let's talk about the incredible enzyme that does the actual building, eukaryotic fatty acid synthase, or FAS. Unlike simpler systems found in bacteria where you have separate enzymes for each step, in complex organisms like humans, FAS isn't a collection of separate enzymes. It's a single, massive, multifunctional protein. Specifically, it's a homodimer, meaning it's made of two identical, large polypeptide chains.
Two identical units working together.
Yes. And each of these units contains 6 different enzymatic domains, plus a special 7th domain called the acyl carrier protein, or ACP domain. This allows it to act as a highly efficient integrated assembly line, adding 2 carbon units derived from malonyl-CoA to the carboxyl end of a growing fatty acid chain. A crucial component of this integrated assembly line is a molecule called 4-meaning-phosphopantothene. It's actually derived from pantothenic acid, vitamin B5.
Vitamin B5.
Yeah. This molecule acts like a flexible swinging arm within the FAS enzyme complex. It carries the growing acyl units on its terminal thiol group, -SH, effectively presenting them sequentially to each catalytic domain. It ensures every step happens efficiently without the intermediates diffusing away. It's also, incidentally, a component of coenzyme A itself.
This enzyme is truly a biological marvel, a whole assembly line packed into one. It's incredibly efficient because it keeps all the different steps and intermediates contained and moving seamlessly, like a tiny molecular factory.
It truly is. Let's walk through the steps of building a common 16-carbon fatty acid palmitate. It's a cyclical process. First, an initial 2-carbon acetyl group derived from acetyl-CoA is loaded onto the ACP domain of FAS. This is done by one of the enzyme activities, malonylacetyl-CoA ACP transacylase. This acetyl group then quickly shifts over to a temporary holding site, specifically a cysteine residue on the condensing enzyme domain, also called 3-ketoacyl ACP synthase.
Okay, so it's parked temporarily.
Exactly. Now, the now empty ACP accepts a 3-carbon malonyl group derived from malonyl-CoA using that same transacylase activity. Now, here's the core reaction. The acetyl group parked on the cysteine residue condenses with the malonyl group tethered to the ACP. Crucially, during this condensation, the carbon dioxide that was initially added by ACC to make malonyl-CoA is released.
Oh, the CO2 comes off again.
Yes, and this decarbokylation is vital. It provides the significant thermodynamic driving force, the free energy, that pushes the entire reaction forward, making the bond formation favorable. The result is a 4-carbon unit, a beta-ketoacyl group, now attached to the ACP domain. This key condensation step is catalyzed by that 3-ketoacyl ACP synthase domain.
Okay, so condensation driven by CO2 release.
Precisely. Following this condensation, the newly formed 4-carbon chain undergoes a series of 3 important modifications, all while still attached to the ACP arm. First, the keto group at the beta carbon is reduced to an alcohol. This step requires an NADPH molecule for the reducing power, the electrons. This is catalyzed by the 3-ketoacyl ACP reductase domain.
First reduction using NADPH.
Correct. Second, a molecule of water is removed from carbons 2 and 3, creating a trans double bond. This dehydration step is done by the 3-hydroxyacyl ACP dehydratase domain. Finally, that newly formed double bond is reduced, saturating the chain. This requires a second molecule of NADPH and is catalyzed by the enoyl ACP reductase domain.
Second reduction also using NADPH.
Yes. So this entire sequence, condensation, reduction, dehydration, and reduction, produces a 4-carbon, fully saturated butyryl group, which remains attached to the flexible ACP arm. This entire cycle then essentially repeats 5 more times.
5 more times, wow.
Yes. Each time, the growing fatty acid chain, now 4 carbons longer than before, is transferred from the ACP to that cysteine holding site. A new 3-carbon malonyl group attaches to the ACP. Then the condensation occurs, followed by the 3 modification steps. Reduction, dehydration, reduction. With each complete turn of the cycle, 2 more carbons are added at the carboxyl end of the growing chain. Once the fatty acid reaches a length of 16 carbons, forming palmitoyl S-ACP, the process generally stops for this enzyme. A specific enzyme activity within the FAS complex, called palmitoyl thioesterase, cleaves the thioester bond between the palmitoyl group and the ACP, releasing a free, fully saturated 16-carbon palmitate molecule.
And that's the final product of this main pathway.
That's the main product. And it's important to understand, really nail this down. All the carbons in this final palmitate molecule, except for the initial 2 from the original acetyl-CoA, which end up at the methyl end, have passed through malonyl-CoA at some point.
Ah, that really highlights why ACC is the bottleneck.
Exactly. It underscores why that initial ACC reaction, making malonyl-CoA, is the key regulatory and overall rate-limiting step for the entire process.
That's an astonishingly precise and efficient assembly line, and it makes perfect sense why that initial ACC step is so tightly controlled. Now, you mentioned NADPH is crucial for all those reduction steps. Where does your body get all that reducing power? Seems like a lot is needed.
You're absolutely right. Palmitate synthesis is quite energy intensive in terms of reducing power. It requires a total of 14 molecules of NADPH for every 1 molecule of palmitate synthesized.
14.
Yeah, it's significant. The primary source of this reducing power, providing roughly half of what's needed, is the pentose phosphate pathway, or PPP. You might remember that pathway.
Right, makes NADPH and precursors for nucleotides.
Exactly. For every molecule of glucose 6-phosphate that enters the oxidative phase of the PPP, 2 molecules of NADPH are produced, so that's a major contributor. Another significant cytosolic source of NADPH comes from the action of cytosolic malic enzyme, also called NADP plus dependent malate dehydrogenase. This enzyme converts malate to pyruvate, and in doing so, it oxidizes malate and uses NADP plus as the electron acceptor, producing NADPH and also releasing CO2.
Okay, malic enzyme. Where does the malate come from?
Good question. And to connect the dots even further, that malate itself can be formed from the reduction of oxaloacetate, or OAA, by a cytosolic enzyme that uses NADH, typically generated during glycolysis. And that OAA, as we discussed earlier with the citrate shuttle, can come from the cleavage of citrate that was transported out from the mitochondria.
Wow, it really loops back.
It does. So you see, it's a beautifully integrated system. It shows how the metabolism of glucose, through both glycolysis and the PPP, is intimately linked to the synthesis of fatty acids, providing both the carbon building blocks via citrate and the necessary reducing power via NADPH, all working in concert to manage your energy.
It's truly a beautiful example of how different metabolic pathways are so elegantly intertwined, all working together in perfect harmony to accomplish a massive task like building fat from scratch. It's not isolated at all.
Indeed. Now, while palmitate, 16 carbons, is the primary product of the main FAS enzyme, your body isn't limited to just that size or saturation level. Palmitate can be further elongated by adding additional 2-carbon units, again derived from malonyl-CoA, to its carboxylate end. This elongation process primarily takes place in the smooth endoplasmic reticulum, or SER, and involves a separate set of enzymes, not the multifunctional FAS complex itself. Malonyl-CoA again acts as the 2-carbon donor, and NADPH supplies the reducing power for the reduction steps involved.
So you can make longer chains if needed.
Yes, and your brain in particular has a remarkable capability to produce very long-chain fatty acids, or VLCFA, which are over 22 carbons long. These are absolutely essential components of specific brain lipids like sphingolipids. Beyond just elongation, your body can also introduce cis double bonds into these fatty acids through a process called desaturation, which also occurs mainly in the SER. This is a complex process involving enzymes called fatty acyl-CoA desaturases. These enzymes require molecular oxygen, O2, NADH, and a sophisticated electron transport system involving cytochrome B5 and an FAD-linked reductase.
So it needs oxygen and NADH. Interesting.
Yes. The first double bond is typically inserted between carbons 9 and 10 of the fatty acyl-CoA chain. This leads to the formation of common monounsaturated fatty acids like palmitoleic acid, 16.1 delta 9 from palmitate, and oleic acid, 18.1 delta 9 from stearate, 18.0. Further desaturation reactions, often coupled with additional elongation steps, can then create various polyunsaturated fatty acids. However, as we mentioned earlier, a critical limitation for humans, and this is worth repeating, is that while we have desaturases that can introduce double bonds at carbons 9, 6, 5, and 4 relative to the carboxyl end, we lack the enzymes needed to introduce double bonds beyond carbon 9, specifically from carbon 10 all the way to the omega end of the chain.
And that's why linoleic and alpha-linolenic are essential.
That is the profound biochemical reason why omega-6 linoleic acid and omega-3 alpha-linolenic acid are nutritionally essential. We simply cannot synthesize them from scratch or from other common fatty acids, making their dietary intake indispensable for health.
So your body is an incredible fatty acid factory, capable of building and customizing these molecules to a large extent, but it absolutely relies on your diet for those truly essential ones, with double bonds near the omega end. It's a powerful reminder that while our bodies are incredibly capable, they're not entirely self-sufficient in every single aspect.
Absolutely. A crucial partnership between internal capability and external supply. Okay, so once fatty acids are synthesized or obtained from the diet, they generally need to be stored for later use. This is where triacylglycerols, or TAGs, come in. They are the body's ultimate, most efficient storage form for fat energy. Imagine a glycerol molecule. It's a simple 3-carbon alcohol. Mono, dia, and triacylglycerols consist of 1, 2, or 3 fatty acid molecules, respectively, attached to this glycerol backbone through ester bonds formed with the hydroxyl groups of glycerol and the carboxyl groups of the fatty acids.
Ester bonds, okay.
This esterification process converts the fatty acids' negatively charged carboxyl groups into neutral esters. This is why TAGs are often referred to as neutral fats. They lack that charge. Interestingly, the physical state depends on the fatty acids. If an acylglycerol is solid at room temperature, we generally call it a fat. If it's liquid, we call it an oil.
Like butter versus olive oil.
Exactly. That difference often comes down to the degree of saturation and chain length of the constituent fatty acids. Now, the 3 fatty acids attached to a single TAG molecule are usually not identical. There's a typical pattern. The fatty acid esterified to carbon 1 of the glycerol backbone is often saturated. The one on carbon 2 is usually unsaturated, and the one on carbon 3 can vary, being either saturated or unsaturated.
Interesting pattern. Does that affect function?
It certainly affects the physical properties. That arrangement, particularly the presence of unsaturated fatty acids with their kinks, significantly influences the TAG's melting temperature and overall fluidity, making the mixture less likely to solidify. In terms of storage and function, because TAGs are extremely hydrophobic, only very slightly soluble in water, they naturally coalesce within your white adipocytes, your fat cells, to form large, essentially anhydrous oily droplets in the cytosol.
Anhydrous meaning without much water.
Exactly. Stored almost purely as fat, making them incredibly energy dense. These cytosolic lipid droplets represent the vast majority of your body's stored energy reserve. And while we mainly think of white fat for energy storage, it's worth remembering that TAGs stored in brown adipocytes serve a different primary purpose, generating heat through non-shivering thermogenesis, which is particularly important in infants and hibernating animals.
So your fat cells are essentially highly efficient, super compact fuel tanks, packing away energy in its most dense and long-lasting form. That makes perfect sense for long-term energy storage.
Absolutely. Now, to actually synthesize these TAGs, a molecule called glycerol 3-phosphate is absolutely crucial. It serves as the initial acceptor of the fatty acids during synthesis.
The backbone they attach to.
Precisely, the initial backbone. There are 2 primary ways your body produces this essential glycerol 3-phosphate. In both the liver, which is a major site of TAG synthesis for export, and in adipose tissue, glycerol 3-phosphate can be made directly from glucose. This involves glucose undergoing the initial steps of glycolysis to produce dihydroxyacetone phosphate, or DHAP. DHAP is then reduced by an enzyme called glycerol 3-phosphate dehydrogenase, using NADH to form glycerol 3-phosphate.
So linked directly to glucose breakdown.
Yes, but there's a second pathway, and this is a key distinction. It's found only in the liver, not in adipose tissue under normal conditions. The liver possesses an enzyme called glycerol kinase.
Kinase adds a phosphate.
Exactly. Glycerol kinase can directly phosphorylate free glycerol, using ATP, to form glycerol 3-phosphate. Adipose tissue generally lacks significant glycerol kinase activity, and this leads to a crucial physiological point. The main glucose transporter in your adipocytes, called GLUT4, is insulin-dependent.
Meaning insulin is needed for glucose to get into fat cells easily.
Correct. This means that when plasma glucose levels are low and insulin is low, as in a fasted state, adipocytes have only a very limited ability to take up glucose, and therefore a limited ability to synthesize glycerol phosphate from glucose. Consequently, because they also lack glycerol kinase, they cannot produce new tags from scratch under these conditions. This elegantly ensures that significant fat storage from glucose is coupled to a state of energy abundance, signaled by insulin.
That's a powerful regulatory point. It explains why your fat cells rely on insulin to even begin storing fat from glucose effectively. It's another layer of control that ensures energy is stored precisely when it's plentiful, not when it's scarce.
Indeed. It prevents a futile cycle of breaking down fat and immediately resynthesizing it from glucose in the same cell when energy is needed elsewhere. Now, before any free fatty acid can be incorporated into tags or participate in virtually any other metabolic pathway like oxidation, it must first be activated. This means it's converted into its energized form where it's linked to coenzyme A through a high-energy thioester bond.
Making fatty acyl-CoA.
Exactly. Fatty acyl-CoA. This activation reaction is catalyzed by a family of enzymes called fatty acyl-CoA synthetases, sometimes known as thiokinases. This step requires ATP. Once you have the activated fatty acyl-CoAs and the glycerol 3-phosphate backbone, the actual tag synthesis pathway involves 4 sequential reactions, occurring mainly in the ER and mitochondria. First, 2 fatty acids, as fatty acyl-CoAs, are added sequentially to glycerol 3-phosphate, forming phosphatidic acid. Then, the phosphate group is removed by a phosphatase, leaving diacylglycerol. DAG. Finally, a third fatty acyl-CoA is added to the DAG to form the final triacylglycerol molecule.
Stepwise addition to the glycerol backbone.
Precisely. Now, what happens to these newly synthesized triacylglycerols? It differs between the liver and adipose tissue. In white adipose tissue, the tags are primarily stored right there as depot fat within those large anhydrous droplets inside the fat cells. This is essentially your body's ready to mobilize long-term fuel reserve, just patiently waiting. In a healthy individual, the liver actually stores very little tag itself. Instead, most of the tag synthesized in the liver is efficiently exported.
The chip step.
It ships it out. It's packaged together with other lipids like cholesterol and phospholipids and specialized proteins called apolipoproteins to form particles known as very low-density lipoproteins, or VLDLs. These nascent VLDL particles are secreted directly from the liver into your bloodstream. There, they mature and function to deliver these endogenously derived lipids, meaning fats made by your own body, mainly the liver to peripheral tissues throughout your body, like muscle and adipose tissue.
Okay, so VLDL carries fats made by the body.
Yes, primarily made by the liver. This contrasts sharply with chylomicrons, which are formed in the intestine and are the primary vehicles for transporting dietary or exogenously derived lipids absorbed from your food.
So your liver acts like a highly sophisticated shipping company, packaging and distributing fats that synthesize itself for delivery throughout the body. It's an incredible distribution system, carefully differentiating between incoming dietary fats and fats it just produced. Keeps everything organized.
Very much so. It's central to lipid homeostasis.
Now that we've journeyed through how your body constructs and stores these vital fats, let's flip the coin and explore how it unleashes that stored energy through fat mobilization and fatty acid oxidation. This is where the true power of fat as fuel truly comes to life, isn't it?
You're absolutely right. Triacylglycerols, those vast fat reserves stored away in your white adipose tissue, are truly your body's major fuel storage reserve. They provide incredibly concentrated stores of metabolic energy, and there are a couple of key reasons for this efficiency. First, fatty acids are highly reduced molecules, meaning they have many carbon-hydrogen bonds capable of releasing a lot of energy when they're oxidized, broken down. Second, as we mentioned, they are largely anhydrous, stored without associating with much water, unlike glycogen, which packs a significant amount of water along with it, making it bulkier and less energy-dense per gram.
Right, less weight for the same energy.
Exactly. To give you a sense of their efficiency, the complete oxidation of fatty acids all the way to carbon dioxide and water yields about 9 kilocalories per gram of fat. Compare that to protein or carbohydrates, both of which yield only about 4 kilocalories per gram when fully metabolized. It's more than double the energy density.
This is a profound insight into your body's design. It's why you can physically keep going for hours, maybe even days, on your fat reserves, while your glycogen stores would be depleted much, much faster, often within hours. It's the ultimate long-distance fuel.
It truly is the preferred long-term storage solution. Now, tapping into these vast reserves requires the controlled release of free fatty acids and glycerol from the stored tags. This process is called lipolysis, which literally means fat breakdown, and it's carried out by a series of specialized enzymes called lipases within the adipocyte. This process is initiated by an enzyme called an adipose triglyceride lipase, or ATGL. ATGL specifically removes the first fatty acid, typically from the C3 or C2 position, leaving a diacylglycerol. This diacylglycerol then becomes the preferred substrate for another critical enzyme called hormone-sensitive lipase, or HSL. HSL removes the second fatty acid, usually from the C1 position, resulting in a monoacylglycerol. Finally, an enzyme called monoacylglycerol (G lipase) acts on the monoacylglycerol, releasing the third and final fatty acid, leaving just free glycerol.
So a cascade of lipases chewing away at the tag.
Precisely. And the regulation of hormone-sensitive lipase, HSL, is absolutely central to controlling the rate of fat mobilization. HSL is most active when it's phosphorylate. This phosphorylation is carried out by a protein kinase A, or pKa, which, as we know, is a CAMP-dependent protein kinase.
CAMP again, linking to hormones.
Exactly. Cyclic AMP levels rise inside the adipocyte when crucial signaling molecules like catecholamines, epinephrine, and norepinephrine, your body's stress hormones, bind to specific beta-adrenergic receptors on the fat cell's membrane. This binding activates adenylate cyclase, which makes CAMP from ATP. The signaling cascade is remarkably similar to how glycogen phosphorylase, the enzyme responsible for breaking down stored glycogen in liver and muscle, is activated. And what's truly ingenious here is the reciprocal regulation we keep seeing. When this CAMP-mediated cascade is activated, signaling that the body urgently needs fuel, fatty acid synthesis, controlled by ACC, is simultaneously inhibited, while tag degradation, driven by HSL, is activated.
Smart. Don't make and break at the same time.
Your body wisely avoids futile cycling. Conversely, when plasma insulin levels are high, typically after a meal signaling energy storage is appropriate, HSL is dephosphorylated by protein phosphatases and becomes much less active, essentially signaling, hold on to the fat, store it. Insulin also plays a role by suppressing the expression of the initiating lipase, ATGL. Another key player in regulating access is a protein called paralipin. This protein coats the surface of the fat droplets within adipocytes. Normally, paralipin acts as a barrier, limiting HSL's access to the stored tag.
A protective coating.
Yes, but when pKa is activated by hormones like epinephrine, it phosphorylates paralipin. This phosphorylation causes paralipin to change its shape, allowing HSL to translocate from the cytosol and effectively bind to the surface of the lipid droplet, initiating the breakdown process.
So in a fight-or-flight situation, or when blood sugar drops and glucagon rises, your body essentially shouts, release the fat. And insulin is the countersignal, the whisper that says, hold on to it, we're in a fed state. It's an incredibly elegant system for energy allocation and survival, really fine-tuned.
Exactly. Now, once glycerol is released during tag degradation, what happens to it? As we touched on, your adipocytes, the fat cells themselves, can't actually metabolize it effectively because they lack significant amounts of the enzyme glycerol kinase. So this released glycerol diffuses out of the fat cell, travels through the bloodstream primarily to your liver, which does possess abundant glycerol kinase.
The liver picks it up.
The liver picks it up. In the liver, glycerol kinase converts it back to glycerol 3-phosphate using ATP. From there, it has a few important fates. It can either be used to reform new tags within the liver, which might then be exported as VLDL, or it can be converted via glycerol 3-phosphate dehydrogenase to dihydroxyacetone phosphate, DHAP. From DHAP, it can then enter glycolysis for immediate energy production within the liver, or, crucially, it can be shunted into gluconeogenesis.
Making new glucose.
Yes, allowing the liver to make new glucose from this fat-derived component, which is particularly important during fasting to maintain blood glucose levels for the brain and red blood cells.
This is a key insight.
Yeah.
Your fat cells release glycerol, which they can't use themselves, but your liver can cleverly convert it into new glucose for the rest of the body. It really highlights the liver's central adaptive role in managing all energy resources during different metabolic states. Very versatile organ.
It absolutely is. As for the free fatty acids, FFAs, released from adipocytes, they also move out of the fat cell, bind non-covalently but tightly to serum albumin in the blood, and are transported to other tissues throughout the body. Particularly active tissues like skeletal muscle, cardiac muscle, and the liver readily take up these FFAs. Once inside these cells, they are activated to their CoA derivatives, fatty acyl-CoAs, and then undergo oxidation for energy, primarily within the mitochondria. However, there are important tissue limitations to remember. Red blood cells cannot use FFAs for fuel at all because they completely lack mitochondria.
No mitochondria, no beta-oxidation.
Correct. And perhaps surprisingly to some, your brain does not use circulating fatty acids for energy to any appreciable extent. Under normal or even fasting conditions, it relies almost exclusively on glucose, or as we'll discuss soon, ketone bodies when glucose is scarce. What's also quite intriguing and maybe counterintuitive is that a significant portion, perhaps over 50% of the fatty acids released from adipose tags during lipolysis are actually reesterified back to glycerol-3-phosphate right there within the white adipose tissue itself, rather than being released into the circulation.
They get recaptured. Why?
Yes. This happens via a process called glyceroneogenesis, which is essentially an abbreviated version of gluconeogenesis occurring within the adipocyte itself. Pyruvate, dinyl-OAA, dilyl-PAP, dilyl-DHAP, glycerol-3-phosphate. This pathway helps to regulate and decrease the overall flux of FFAs leaving the adipose tissue and entering the plasma. This is physiologically important because chronically elevated plasma FFA levels are strongly associated with the development of insulin resistance in peripheral tissues, a hallmark of conditions like type 2 diabetes and obesity. So it's a sort of internal buffering system.
It's a remarkably complex and dynamic dance. Not all tissues can use fat directly. The brain has its preferences, and even fat cells have this ingenious way to catch and restore some of their released fuel if it's not immediately needed, possibly to protect other tissues. It really underscores the body's incredible efficiency and multiple layers of intricate regulation.
Absolutely. This complex interplay brings us squarely to the primary way fatty acids are actually broken down to harvest their stored energy. The process known as fatty acid beta-oxidation. This is the major catabolic pathway for fatty acids, occurring primarily inside the mitochondria of most cells. The core process involves the successive removal of 2 carbon fragments from the carboxyl end of the fatty acyl-CoA molecule. Each round generates 1 molecule of acetyl-CoA, 1 molecule of NADH, and 1 molecule of FADH2. But as we alluded to, there's a significant hurdle for long-chain fatty acids, typically those with 14 carbons or more, to even get into the mitochondria where the beta-oxidation machinery resides.
The transport problem again.
Exactly. Once a long-chain fatty acid enters a cell's cytosol, it's first activated to its CoA derivative, fatty acyl-CoA, by enzymes called long-chain fatty acyl-CoA synthetases, which are primarily located on the outer mitochondrial membrane and the ER. The challenge is that beta-oxidation itself takes place deep inside, in the mitochondrial matrix, the innermost compartment. And the inner mitochondrial membrane is effectively impermeable to CoA and its derivatives, like fatty acyl-CoA.
So it's stuck outside the main factory.
Precisely. To overcome this barrier, a specialized carrier system known as the carnitine shuttle is absolutely required to transport these long-chain acyl groups across the inner membrane into the matrix. This carnitine shuttle is not just a simple transporter, it's also the primary rate-limiting transport process for the oxidation of long-chain fatty acids, making it a critical point of regulation.
Think of carnitine as the vital ferryman, tirelessly bringing the long fatty acids across the mitochondrial river, that inner membrane, to where the energy production happens. Without this ferryman, those long fatty acids are essentially stuck on the wrong side, unable to be burned for fuel.
That's a perfect analogy. Let's detail exactly how this crucial translocation works. It involves 3 key components. First, on the outer mitochondrial membrane, an enzyme called carnitine palmitoyl transferase I, or CPTI, catalyzes the transfer of the long-chain acyl group from CoA to the hydroxyl group of carnitine. This forms acylcarnitine and regenerates free CoA in the cytosol, or intermembrane space.
So fatty acid hops from CoA onto carnitine.
Exactly. Second, this newly formed acylcarnitine molecule is then transported across the impermeable inner mitochondrial membrane into the matrix by a specific protein called the carnitine acylcarnitine translocase. This transporter works like an antiporter. It simultaneously moves 1 molecule of acylcarnitine in while moving 1 molecule of free carnitine out into the intermembrane space.
An exchange system.
A direct exchange. Finally, once the acylcarnitine is inside the mitochondrial matrix, a second enzyme, carnitine palmitoyl transferase 2, or CPT2, which is located on the matrix side of the inner mitochondrial membrane, catalyzes the reverse reaction. It transfers the acyl group from carnitine back onto a molecule of CoA that's present within the matrix. This regenerates the fatty acyl-CoA inside the matrix, ready for beta-oxidation, and frees up the carnitine molecule. That free carnitine is then shuttled back out by the translocase to pick up another fatty acid.
A neat cycle to get the fatty acid across.
A very neat and essential cycle. And crucially, this system incorporates a brilliant layer of reciprocal regulation involving malonyl-CoA.
Malonyl-CoA from fat synthesis.
the very same key intermediate from fatty acid synthesis. Malonyl-CoA, which builds up in the cytosol when synthesis is active, like after a high-carb meal when insulin is high, acts as a potent inhibitor of CPCI, the enzyme on the outer membrane. This is a masterstroke of metabolic control because it effectively prevents newly synthesized fatty acids, like palmitate, from immediately entering the mitochondria for degradation at the same time the cell is actively trying to synthesize and store fat.
That is truly genius. Your body has a built-in do not disturb sign. If you're busy making fat in the cytosol, you're actively preventing it from being burned in the mitochondria. This level of coordinated control between opposing pathways is astounding.
It really is. It prevents a wasteful, futile cycle. It's also worth noting that even tissues like muscle, although they don't typically synthesize large amounts of fatty acids for storage themselves, contain a specific mitochondrial isozyme of ACC called ACC2. The malonyl-CoA produced by ACC2 in muscle mitochondria likely serves primarily to regulate CPTI activity locally, thereby controlling the rate of fatty acid oxidation according to the muscle's energy status, rather than contributing significantly to fat synthesis. The liver has both ACC isozymes. Furthermore, fatty acid oxidation within the mitochondrion is also subject to feedback regulation by the ratio of acetyl-CoA to free-CoA. A high ratio signals abundant energy from oxidation and can slow down the final thiolase step of beta-oxidation, effectively putting the brakes on further breakdown when the products aren't needed immediately by the TCA cycle.
Multiple layers of control, both transport and enzyme activity.
Absolutely. Now, regarding carnitine itself, the ferritin molecule, where does it come from? Your body can obtain it directly from your diet, where it's particularly abundant in meat products. It can also synthesize it internally from the essential amino acids, lysine and methionine. This synthesis process, however, primarily occurs in the liver and kidneys. Tissues with very high energy demands that rely heavily on fatty acid oxidation, like skeletal muscle and cardiac muscle, cannot synthesize carnitine themselves. They are entirely dependent on taking up carnitine supplied either from endogenous synthesis elsewhere or from the diet distributed via the bloodstream. In fact, skeletal muscle alone accounts for the vast majority, something like 97% of all the carnitine stored in your body.
Say muscle really hoards it.
It does, because it needs it constantly. Unfortunately, carnitine deficiencies can occur and have severe consequences, greatly reducing the ability of these crucial tissues to use long-chain fatty acids as fuel, forcing them to rely more heavily on glucose. Primary carnitine deficiency is typically caused by genetic defects in a specific plasma membrane transporter protein, OCTN2, that's responsible for carnitine uptake into cells, particularly in muscle, heart, and kidney. This leads to excessive carnitine loss in the urine and dangerously low carnitine levels in tissues. Treatment usually involves lifelong high-dose carnitine supplementation. Secondary carnitine deficiency, on the other hand, is more common and primarily results from other underlying metabolic problems. Often, it arises from defects within the fatty acid oxidation pathway itself, which lead to the accumulation of abnormal acylcarnitine intermediates inside cells. These abnormal acylcarnitines can then be excreted in the urine, effectively trapping and depleting the body's pool of free carnitine. This situation can also arise in patients with severe liver disease due to decreased carnitine synthesis, or in those taking certain medications like the anti-seizure drug valproic acid, which is known to impair the renal reabsorption of carnitine.
So either not getting it in or losing too much of it.
Essentially, yes. And there are also specific genetic deficiencies related to the CPT enzymes themselves, CPTI and CPT2. CPTI deficiency primarily affects the liver, causing a profound inability to use long-chain fatty acids for fuel. This severely impairs the liver's capacity for gluconeogenesis during periods of fasting, because glucose synthesis is an energy-demanding process that relies heavily on the ATP generated from fatty acid oxidation. This impairment can lead to episodes of severe hypoglycemia, low blood sugar, lethargy, seizures, coma, and tragically, even death if not managed promptly. CPT deficiency is generally more common and can manifest in several forms, affecting the liver, cardiac muscle, and skeletal muscle. The most common form, which is generally less severe and often presents in adolescence or adulthood, primarily affects skeletal muscle. It typically presents as episodes of muscle weakness, muscle pain, myalgia, and often myoglobinuria, the release of muscle protein into the urine, turning it dark, especially following prolonged exercise, fasting, or illness.
So exercise triggers the symptoms because the muscles can't burn fat properly.
Exactly. The energy demand outstrips the muscle's ability to oxidize fat. Treatment for the common muscle form of CPT deficiency involves careful management. Avoiding prolonged fasting, adopting a diet that is relatively high in carbohydrates and low in long-chain fats, and often supplementing with medium-chain triacylglycerols, MCTs.
Why MCTs?
Good question. It's worth noting that shorter-chain fatty acids, specifically those with 12 carbons or less, medium-chain and short-chain, actually have an easier time entering the mitochondria. They can cross the inner mitochondrial membrane without the aid of carnitine or the CPT system.
They don't need the ferryman.
They don't need the ferryman. Once inside the mitochondrial matrix, they are activated to their CoA derivatives by specific medium or short-chain acyl-CoA synthetases located there, and then they readily undergo beta-oxidation. This is a key reason why medium-chain fatty acids, MCFAs, like those abundant in human milk or commercially available MCT oil, often derived from coconut oil, are so easily and rapidly oxidized. Their entry into the mitochondria is not dependent on CPTI, meaning malonyl-CoA does not inhibit their breakdown either. This makes them a useful energy source in conditions where long-chain fat oxidation is impaired.
That makes sense. A way to bypass the bottleneck.
Precisely. Okay, so once the fatty acyl-CoA is inside the mitochondrial matrix, let's detail the core beta-oxidation reactions themselves. It's a repeating cycle, often called the beta-oxidation spiral. This sequence consists of 4 distinct enzymatic reactions, all focusing on modifying the bond between the alpha-carbon C2 and the beta-carbon C3 relative to the carboxyl N, C1. Each complete cycle achieves 2 main things. It shortens the fatty acyl-CoA chain by 2 carbons, releasing those 2 carbons as 1 molecule of acetyl-CoA, and it generates reducing equivalents in the form of 1 molecule of FADH2 and 1 molecule of NADH.
4 steps per cycle.
4 steps. First, there's an oxidation step catalyzed by acyl-CoA dehydrogenase. This enzyme introduces a trans double bond between the alpha and beta carbons, and the electrons removed are transferred to FAD, producing FADH2. There are actually multiple acyl-CoA dehydrogenases with different specificities for short, medium, long, and very long-chain fatty acids. Second, a hydration step occurs, catalyzed by a noyl-CoA hydratase. This enzyme adds a molecule of water across that newly formed double bond, creating a hydroxyl group on the beta carbon. Third, a second oxidation step takes place, catalyzed by beta-hydroxycyl-CoA dehydrogenase. This enzyme oxidizes the hydroxyl group on the beta carbon to a keto group, and the electrons removed are transferred to NAD plus TESI, producing NADH. Finally, the fourth step is a thiolytic cleavage, catalyzed by beta-ketothiolase, or simply thiolase. This enzyme uses a molecule of free coenzyme A to break the bond between the alpha and beta carbons. This releases the first 2 carbons as acetyl-CoA and leaves behind a fatty acyl-CoA molecule that is now 2 carbons shorter than the original.
And that shorter fatty acyl-CoA goes back into the cycle.
Exactly. That shortened fatty acyl-CoA is now the substrate for the next round of the 4-step cycle. For saturated fatty acids with an even number of carbon atoms, which are the most common, this 4-step cycle repeats N2 1 times, where N is the total number of carbons in the original fatty acid. Each round yields 1 acetyl-CoA, 1 FADH2, and 1 NADH. The final cycle is unique because the thiolase step cleaves a 4-carbon beta-ketoacyl-CoA into 2 molecules of acetyl-CoA.
Okay, so the last round gives 2 acetyl-CoAs.
Correct. Now, what happens to these products? The acetyl-CoA produced can then enter the TCA cycle, Krebs cycle, right there in the mitochondrial matrix for further complete oxidation to carbon dioxide and water, generating much more ATP. Or, particularly in the liver during periods of high-fat breakdown, acetyl-CoA can be diverted towards hepatic ketogenesis, the synthesis of ketone bodies, which we'll discuss next. The NADH and FADH2 produced during each cycle are absolutely crucial. They are rapidly reoxidized by the mitochondrial electron transport chain. NADH donates its electrons to complex I, and FADH2 donates its electrons via electron-transferring flavoprotein and ETFQ oxidoreductase, ultimately to coenzyme Q, ubiquinone. The flow of these electrons down the chain drives the pumping of protons, creating the proton gradient that ultimately powers ATP synthase to make the vast majority of your cellular ATP via oxidative phosphorylation. Interestingly, as another point of integration, the acetyl-CoA produced by beta-oxidation also acts as a positive allosteric effector of pyruvate carboxylase, the enzyme that converts pyruvate to oxaloacetate. This provides a crucial link connecting high rates of fatty acid oxidation, generating lots of acetyl-CoA, to the stimulation of gluconeogenesis, which requires OAA.
It makes sure the pathways are coordinated. If you're burning lots of fat, maybe you need to make glucose too.
Precisely. It helps replenish TCA cycle intermediates and provides precursors for glucose synthesis when needed.
This is truly a remarkable testament to energy efficiency. Fat is indeed a super dense fuel. It's like unlocking an enormous, efficient power source right within your own cells. The ATP yield must be huge.
It absolutely is. The energy yield from fatty acid beta-oxidation is incredibly high compared to glucose. Let's take our example of palmitoyl-CoA, which has 16 carbons. Its complete beta-oxidation produces 8 molecules of acetyl-CoA, 7 molecules of FADH2, and 7 molecules of NADH. If you then run those 8 acetyl-CoA molecules through the TCA cycle and put all the resulting NADH and FADH2 from both beta-oxidation and the TCA cycle through the electron transport chain, the theoretical maximum AQI yield is around 108 ATP molecules just from the reoxidation of the reducing equivalents and GTP from the TCA cycle. However, standard biochemistry textbooks often use slightly different pO ratios, leading to varying final numbers. A commonly cited total yield for the complete oxidation of one molecule of palmitate, starting from the free fatty acid, is around 106 net ATP molecules, after accounting for the initial 2 ATP equivalents used for activation in the cytosol. Older calculations might give numbers like 129 or 131 ATP before subtracting activation cost, depending on the assumptions used.
Wow, over 100 ATP from just one 16-carbon fat molecule, compared to maybe 30, 32 from one glucose.
Exactly. It's far more efficient on a per molecule or per gram basis than carbohydrate breakdown. And if you compare the overall processes of fatty acid synthesis and degradation side by side, you'd observe that they are distinct pathways, occurring in different cellular compartments, cytosol versus mitochondria, utilizing different enzymes, different electron carriers, NADPH for synthesis, NAD plus FAD for oxidation, and different key intermediates. Malonyl-CoA is unique to synthesis. This spatial and biochemical separation is a key design principle that allows them to be independently and reciprocally regulated. When one pathway, like synthesis, is active, the other, degradation, is generally inhibited, preventing wasteful conflict.
Brilliant design for control.
Indeed. Now let's consider some important clinical implications of defects within this beta-oxidation process itself. We mentioned CPT deficiencies, but defects in the dehydrogenases are also significant. Medium-chain fatty acyl-CoA dehydrogenase deficiency, or MCAD deficiency, is probably the most common inherited disorder of beta-oxidation. It's an autosomal recessive condition affecting roughly 1 in 14,000 births, with a higher prevalence in individuals of Northern European descent. As the name suggests, this deficiency means the body has a significantly decreased ability to oxidize fatty acids with chain lengths between 6 and 10 carbons. These medium-chain fatty acyl-CoAs and their metabolites then tend to accumulate within mitochondria and spill out, leading to characteristic patterns of dicarboxylic acids and acylglycines that can be detected in urine, especially during periods of metabolic stress.
So the medium chains build up because they can't be broken down properly.
Correct. Clinically, MCAD deficiency often presents in infancy or early childhood, typically triggered by a period of fasting or illness that increases reliance on fat metabolism. The key manifestations are episodes of severe hypoketotic hypoglycemia.
Hypoglycemia because they can't use fat well and rely too much on glucose.
Exactly.
And hypoketotic because they can't make enough acetyl-CoA from fat to produce ketones.
Precisely. They lack the acetyl-CoA substrate needed for significant ketone body synthesis in the liver. This combination can lead to lethargy, vomiting, seizures, coma, and unfortunately, sudden infant death if not recognized and treated. The primary cornerstone of management for MCAD deficiency is strict avoidance of prolonged fasting and ensuring adequate caloric intake, especially during illness. Newborn screening programs now routinely test for MCAD deficiency in many countries, allowing for early diagnosis and intervention.
This is a critical example of how a single enzyme deficiency can completely derail your body's energy balance, especially during periods of fasting or stress when fat breakdown is essential. It underscores the precision and reliability required for these metabolic processes to function correctly for survival.
It truly does. Now, not all naturally occurring fatty acids have an even number of carbons. While less common, odd-chain fatty acids are found in the diet, for example, from dairy fats, certain plants, and are also produced by gut bacteria. The beta-oxidation of fatty acids with an odd number of carbons proceeds exactly like that of even-chain fatty acids, removing 2 carbon units as acetyl-CoA in each cycle until the final 3 carbons remain. This unique 3-carbon product is called propionyl-CoA. Propionyl-CoA is also generated during the metabolism of certain amino acids like valine, isoleucine, methionine, threonine, and cholesterol side chains.
So propionyl-CoA comes from a few sources. What happens to it?
The metabolism of propionyl-CoA involves a distinct 3-step pathway that converts it into succinyl-CoA, which can then enter the TCA cycle. First, propionyl-CoA is carboxylated, meaning CO2 is added, to form D-methylmalonyl-CoA. This reaction is catalyzed by the enzyme propionyl-CoA carboxylase, and it absolutely requires the vitamin biotin as a coenzyme to carry the activated CO2, as well as ATP for energy. Second, the D-isomer of methylmalonyl-CoA is converted to its L-isomer by an enzyme called methylmalonyl-CoA racemase. Finally, and this is a fascinating step, the carbons of L-methylmalonyl-CoA undergo an intermolecular rearrangement to form succinyl-CoA. This reaction is catalyzed by the enzyme methylmalonyl-CoA mutase.
And succinyl-CoA is a TCA cycle intermediate?
Correct. Succinyl-CoA can readily enter the TCA cycle for further metabolism. What's particularly significant about this pathway is that the conversion to succinyl-CoA means that the carbons from odd-chain fatty acids, via propionyl-CoA, can ultimately contribute to the net synthesis of oxaloacetate, OA, within the TCA cycle. And OAA is a key precursor for gluconeogenesis.
Ah, so this is the link. Odd-chain fats can be used to make glucose.
Yes, this is the only example where fatty acid oxidation can lead to a net production of glucogenic precursors. Even-chain fatty acids only produce acetyl-CoA, which cannot be used for net glucose synthesis in mammals. So odd-chain fatty acids are unique in being partially glucogenic. Now, the final enzyme in this pathway, methylmalonyl-CoA mutase, is clinically very important because it crucially requires a specific coenzyme form of vitamin B12, specifically deoxydenocylcobalamin. This mutase reaction is one of only 2 enzymatic reactions in your entire body known to absolutely require vitamin B12, the other being methionine synthase.
Only 2 B12 reactions. That's remarkable.
It is, and this has clinical consequences. In patients with vitamin B12 deficiency, often due to pernicious anemia or dietary insufficiency, this pathway is blocked. As a result, both propionate and, more significantly, methylmalonate accumulate and are excreted in the urine, methylmalonic aciduria. Measuring elevated methylmalonic acid is actually a sensitive functional indicator of B12 deficiency. There are also rare inherited forms of methylmalonic acidemia and aciduria caused either by a genetic deficiency in the mutase enzyme itself or by defects in the complex process of converting dietary vitamin B12 into its active coenzyme form. Both types result in a buildup of methylmalonic acid, leading to severe metabolic acidosis, developmental delay, and often serious neurologic manifestations.
So a seemingly small difference in chain length having an odd number of carbons can lead to a whole new metabolic fate, connecting fat to glucose synthesis, and even a relatively common vitamin deficiency like B12 can throw a huge wrench in the works. It's incredible how specific these requirements are and how interconnected our nutritional intake is with core metabolic function.
Absolutely. The body's metabolic machinery is incredibly specific, and its adaptability extends to handling the challenges posed by unsaturated fatty acids during beta-oxidation as well. The presence of naturally occurring cis double bonds in unsaturated fatty acids creates intermediates during beta-oxidation that are unsuitable substrates for one of the standard enzymes, specifically the 2-coupled-3-enoyl-CoA hydratase, which expects a trans double bond.
The kinks cause problems for the standard enzymes.
They do. Consequently, your body requires additional specialized auxiliary enzymes to process these unsaturated fatty acids properly. The exact enzymes needed depend on the position and configuration of the double bonds. If there's a double bond starting at an odd-numbered carbon position relative to the carboxyl end, such as the delta-9 bond in oleic acid, 18.19, beta-oxidation proceeds normally until that double bond is encountered. This creates a 3-cis derivative, which the hydratase can't handle. Here, one additional enzyme is required, an isomerase called 3'2-enoyl-CoA isomerase. This enzyme simply repositions the double bond, converting the 3-cis derivative into the standard 2-trans derivative, which can then be processed normally by the hydratase and the rest of the beta-oxidation cycle.
So just an isomerase needed for single odd-position double bonds.
Yeah, correct. However, for polyunsaturated fatty acids, especially those with double bonds starting at even-numbered carbon positions, like the delta-12 bond in linoleic acid, 18.2912, the process becomes more complex. When beta-oxidation reaches an even-numbered double bond, it ultimately generates a 2'4-dienoyl-CoA intermediate. This requires the action of an NADPH-dependent enzyme called 2'4-dienoyl-CoA reductase, in addition to the isomerase mentioned before. The reductase first reduces one of the double bonds, and then the isomerase acts to get the remaining double bond into the correct position for the hydratase.
More enzymes needed for the more complex structures.
Exactly. The overall result is that because unsaturated fatty acids are already partially oxidized, they have fewer CH bonds than saturated ones due to the double bonds, their complete beta-oxidation actually produces slightly fewer total reducing equivalents. Fewer FADH2 molecules are generated for each double bond present, and yields a little less ATP per carbon compared to their fully saturated counterparts.
Your body is truly incredibly adaptable, though. It has specialized tools and pathways ready to handle those kinks and bends in unsaturated fats, ensuring maximum energy extraction even from these more complex structures. It's like having a universal toolkit designed to handle almost any type of fatty acid fuel.
It does. And the versatility doesn't stop there. Beyond the main mitochondrial beta-oxidation pathway, there's also a significant role for peroxisomal beta-oxidation, particularly for certain types of fatty acids. Very long-chain fatty acids, or VLCFAs, generally defined as those with 22 carbons or longer, primarily undergo an initial round of shortening via beta-oxidation within specialized cellular organelles called peroxisomes before they are fully oxidized in mitochondria.
Why peroxisomes first?
The main reason seems to be that the specific acyl-CoA synthetase enzyme that efficiently activates these extremely long fatty acids for breakdown is primarily located in the peroxis, not the mitochondria, so they have to start their breakdown where they get activated. Once partially shortened by a few cycles of beta-oxidation within the peroxisome, the resulting medium or long-chain fatty acyl-CoA, often linked to carnitine for transport out of the peroxisome, can then diffuse or be transported to a mitochondrion for further complete oxidation via the standard mitochondrial beta-oxidation pathway.
So peroxisomes handle the initial trimming of the really long ones.
Exactly. Now, there's a crucial biochemical difference between peroxisomal and mitochondrial beta-oxidation, particularly in the very first step. In peroxisomes, the initial dehydrogenation step, introducing the double bond, is catalyzed by an FAD-containing enzyme called acyl-CoA oxidase, not an acyl-CoA dehydrogenase like in mitochondria. The FADH2 produced by this oxidase reacts directly with molecular oxygen O2 to produce hydrogen peroxide, H2O2.
Hydrogen peroxide, that sounds dangerous.
It can be, but peroxisomes are well equipped to handle it. Crucially, this direct reaction with oxygen means that the energy from this first oxidation step in the peroxisome is not captured as ATP via the electron transport chain. It's essentially lost as heat. The potentially damaging H2O2 produced is then immediately and safely detoxified, broken down into water and oxygen by another highly abundant enzyme within the peroxisome called catalase. The remaining steps of peroxisomal beta-oxidation, hydration, second oxidation yielding NADH, and thiolytic cleavage are similar to those in mitochondria, although catalyzed by different enzymes or isoforms.
So less ATP generated in peroxisomal oxidation for that first step.
Correct. This pathway has very significant clinical implications when it malfunctions. Genetic defects in the complex process of assembling functional peroxisomes, involving proteins called peroxins, result in a group of severe disorders known as peroxisomal biogenesis disorders, PBDs, the most severe form being Zellweger syndrome. In these conditions, multiple peroxisomal functions are impaired, leading to the toxic accumulation of both VLCFAs and another substrate, phytanic acid, which we'll mention next, in tissues throughout the body, causing profound neurological damage and early death. Another distinct but related X-linked genetic disorder is adrenoleukodystrophy, XALD. This is caused by defects in a specific transporter protein, ABCD1, located in the peroxisomal membrane that is responsible for importing VLCFAs into the peroxisome. In this case, other peroxisomal functions are normal, but the inability to transport and break down VLCFAs leads specifically to their accumulation, particularly in the brain's white matter and the adrenal cortex, causing progressive neurological deterioration and adrenal insufficiency.
This really highlights how absolutely critical these cellular compartments, like peroxisomes and their specific transport systems, are. When the delivery system for specific enzymes or the molecules they act upon breaks down, the consequences can be devastatingly severe and widespread across multiple body systems. It's a stark reminder of the importance of precise cellular organization right down to the molecular level.
Precisely. And the peroxisome handles even more specialized fatty acid structures. There's also a process called peroxisomal alpha oxidation. This pathway is specifically designed to handle certain branched chain fatty acids, the prime example being phytanic acid. Phytanic acid is derived from the metabolism of phytol, a component of chlorophyll found in green vegetables and dairy products from ruminant animals. Phytanic acid cannot be directly processed by the standard beta-oxidation pathway because it has a methyl group attached to its beta carbon, which physically blocks the action of the first enzyme, acyl-CoA dehydrogenase.
The branch gets in the way.
Exactly, so it undergoes alpha oxidation instead. This involves an initial hydroxylation reaction at the alpha carbon C2, catalyzed by an enzyme called phenyl-CoA alpha hydroxylase, PHOI. Then the original carboxyl carbon C1 is removed as carbon dioxide, or formate. The resulting product, which is now one carbon shorter and called pristynol, is oxidized to pristanic acid. Prostanic acid now has a methyl group on its alpha carbon, but its beta carbon is unsubstituted, so it can be activated to pristanoyl-CoA and then undergo normal beta oxidation within the peroxisome.
A clever workaround to deal with the branch.
A very clever metabolic detour. There's a rare but serious autosomal recessive genetic disorder called Refsum disease, which is caused by a deficiency of the peroxisomal Phi-H enzyme needed for alpha oxidation. This leads to the massive accumulation of toxic levels of phytanic acid in the plasma and various tissues, primarily resulting in progressive neurological symptoms like peripheral neuropathy, cerebellar ataxia, and retinitis pigmentosa. Thankfully, treatment for Refsum disease involves strict dietary restriction of phytanic acid sources, which can halt or slow disease progression if started early. It's also worth briefly mentioning one other minor fatty acid oxidation pathway called omega oxidation. This pathway involves oxidation occurring at the omega carbon, the methyl group at the very end of the fatty acid chain opposite the carboxyl group. This process, occurring mainly in the smooth endoplasmic reticulum, generates dicarboxylic acids, acids with carboxyl groups at both ends. Normally, omega oxidation is not a major contributor to overall fat breakdown. However, its activity can be significantly upregulated as a compensatory mechanism in conditions where the primary beta oxidation pathway is limited or overwhelmed, for example, in MCAD deficiency or other conditions leading to an accumulation of fatty acids. The resulting dicarboxylic acids are more water-soluble and can be excreted in the urine.
Your body truly has a specialized pathway, or at least a backup plan, for even the trickiest and most unusually structured fatty acids, demonstrating its incredible metabolic versatility and ability to adapt to diverse fuel types and challenging situations. It's constantly finding ways to manage energy flow.
Absolutely. It's a testament to evolutionary fine-tuning.
All right, let's move on to our final major player in this intricate energy story, the ketone bodies. These are truly fascinating molecules, often described, especially in popular media lately, as your brain's emergency fuel or alternative fuel. What exactly are they, and why do they become so incredibly important during certain metabolic states like fasting or low-carb diets?
Right. The compounds collectively categorized as ketone bodies are actually a trio of small, water-soluble molecules: acetoacetate, 3-hydroxybutyrate, which you might also see called beta-hydroxybutyrate or BHB, and acetone. Now, acetone is typically considered a biologically inert, non-metabolized side product. It's quite volatile and is simply exhaled via the lungs if it builds up. But acetoacetate and 3-hydroxybutyrate are the metabolically active players here. Technically, only acetoacetate is a true ketone, while 3-hydroxybutyrate is a hydroxy acid, but they function together. They are both organic acids.
Okay, acetoacetate and 3-hydroxybutyrate are the main ones.
Yes. Their crucial advantage lies in their unique transportation properties. They are highly soluble in aqueous solutions, like blood plasma. This means, unlike fatty acids or triglycerides, which need complex lipoprotein particles or albumin for transport, ketone bodies do not. They can be easily and rapidly transported, dissolved directly in the blood to virtually any tissue in the body that has mitochondria and the necessary enzymes to use them.
So easy delivery anywhere.
Very easy delivery. They are predominantly produced, almost exclusively synthesized in the liver, specifically within the liver mitochondria. This happens during periods when the rate of fatty acid oxidation in the liver is very high, leading to the production of acetyl-CoA that exceeds the liver's capacity to process it through the TCA cycle. Think prolonged fasting, very low carbohydrate diets, or uncontrolled type 1 diabetes. What's particularly crucial is that these ketone bodies, produced by the liver, are readily taken up and used for energy by most extrahepatic tissues, that is, tissues outside the liver. This includes vital organs like your skeletal muscle, cardiac muscle, which actually prefers ketones and fatty acids over glucose, the intestinal lining, enterocytes, and the renal cortex, kidney. These tissues use ketones in direct proportion to their concentration circulating in the blood. The more ketones available, the more they tend to use them.
So lots of tissues can use them.
Yes. And here's the really profound insight regarding the brain. Your brain, which under normal conditions relies almost exclusively on glucose for its considerable energy needs, can and will adapt over time to use ketone bodies as a major fuel source if their blood levels rise sufficiently, as occurs during prolonged fasting or ketogenic diets. This adaptation is absolutely vital for survival during extended periods without food. By utilizing ketones, the brain significantly reduces its demand for glucose. This helps to spare the body's precious protein stores in muscle from being broken down extensively to provide amino acids for gluconeogenesis, making glucose in the liver. It's a critical glucose-sparing and protein-sparing effect.
A key survival mechanism.
Absolutely. It allows humans to survive for weeks without eating. It's also important, clinically, to remember that disorders of fatty acid oxidation, like MCAD deficiency that we discussed, often present with both hypoketosis and abnormally low level of ketone bodies due to the reduced supply of acetyl-CoA needed for their synthesis, alongside the hypoglycemia that results from the impaired fat burning and increased reliance on glucose. The inability to make ketones exacerbates the danger of fasting in these conditions.
So ketone bodies are like your body's brilliant backup generator, especially crucial for keeping your brain fueled and sharp when glucose runs scarce. It's a remarkable, life-sustaining adaptation forged by evolution.
It absolutely is. Now, let's look more closely at how the liver, and primarily only the liver, synthesizes these ketone bodies in a process called ketogenesis. The primary trigger, as mentioned, is a high rate of fatty acid oxidation, typically occurring during a prolonged fasting or very low carbohydrate intake. In these states, your liver is flooded with free fatty acids that have been mobilized from your adipose tissue stores. Beta-oxidation ramps up dramatically, leading to significantly elevated levels of acetyl-CoA within the liver mitochondria.
Lots of acetyl-CoA building up.
Yes. Now, this high concentration of acetyl-CoA has several important consequences within the liver cell. First, it acts as an allosteric activator of pyruvate carboxylase, the enzyme that converts pyruvate to oxaloacetate, OAA. Second, it tends to inhibit pyruvate dehydrogenase, the enzyme that converts pyruvate into acetyl-CoA. Simultaneously, the very high rate of fatty acid oxidation generates a large amount of NADH, leading to a low NAD plus NADH ratio within the mitochondria. This low ratio favors the reduction of OAA to malate, further decreasing the available pool of OAA.
So OAA gets used for gluconeogenesis and also shifted away to malate.
Exactly. The net result is that the availability of OAA for condensation with acetyl-CoA in the first step of the TCA cycle becomes limited. Even though acetyl-CoA levels are high, the TCA cycle can't keep pace because its starting partner, OAA, is being diverted elsewhere, primarily towards making glucose.
The TCA cycle slows down relative to acetyl-CoA production.
Precisely. This leads to the excess acetyl-CoA being shunted towards an alternative pathway, ketone body synthesis. It's worth noting that acetyl-CoA destined for ketogenesis can also come from the catabolism of purely ketogenic amino acids like leucine and lysine, although fatty acids are the predominant source during fasting.
So it's a brilliant, multi-layered adaptation driven by hormonal signals and substrate availability. When your body urgently needs to make glucose for essential tissues, using OAA, it simultaneously shifts its other main energy strategy towards burning fat heavily and producing ketones from the excess acetyl-CoA, providing an alternative fuel for the brain and other tissues. It's all about optimal resource allocation during times of glucose scarcity.
Exactly. It's a beautifully coordinated metabolic shift. Now, the synthesis pathway for ketone bodies itself within the liver mitochondria begins with the enzymatic condensation of two molecules of acetyl-CoA to form acetoacetyl-CoA. This reaction is catalyzed by the enzyme thiolase, essentially running the last step of beta-oxidation in reverse. Then, a crucial enzyme, mitochondrial HMG-CoA synthase, distinct from the cytosolic version used for cholesterol synthesis, combines this acetoacetyl-CoA with a third molecule of acetyl-CoA to produce a 6-carbon intermediate called 3-hydroxy-3-methylglutaryl-CoA, or HMG-CoA.
HMG-CoA.
This HMG-CoA synthase step is generally considered the rate-limiting and committed step in the synthesis of ketone bodies, and its activity is significantly high primarily, almost exclusively, in the liver mitochondria. This enzyme's presence largely defines the liver's unique capacity for ketogenesis. Next, another enzyme unique to mitochondria, HMG-CoA, cleaves this HMG-CoA molecule. It breaks off one molecule of acetyl-CoA and leaves behind the first ketone body, the 4-carbon molecule acetoacetate.
So HMG-CoA gets split into acetyl-CoA and acetoacetate.
Correct. Acetoacetate is then at a metabolic branch point. It can be released directly from the liver into the bloodstream. Alternatively, within the liver mitochondria, it can be enzymatically reduced to form the second major ketone body, 3-hydroxybutyrate, or BHB. This reduction reaction uses NADH as the electron donor and is catalyzed by the enzyme 3-hydroxybutyrate dehydrogenase. The ratio of 3-hydroxybutyrate to acetoacetate produced by the liver is directly dependent on the mitochondrial NAD plus NADH ratio. When fatty acid oxidation is very high, NADH levels are high, low NAD plus NADH ratio, which strongly favors the reduction of acetoacetate to 3-hydroxybutyrate. So during significant ketosis, BHB is often the predominant ketone body circulating in the blood.
The ratio reflects the liver's redox state.
Precisely. Finally, acetoacetate has one other fate. It is chemically somewhat unstable and can spontaneously, non-enzymatically decarboxylate, losing CO2, to form the third ketone body, acetone. This happens gradually in the blood. As we mentioned, acetone is volatile, largely non-metabolized, and simply diffuses out of the blood into the lungs and is exhaled, sometimes giving a characteristic sweet or fruity odor to the breath in individuals with very high ketone levels, ketoacidosis.
So mainly acetoacetate and BHB are the fuels, acetone is mostly waste.
Exactly. Now, how do the peripheral tissues actually use these ketone bodies delivered by the blood? This process is called ketolysis. While the liver consistently synthesizes low levels of ketones even in the fed state, their production dramatically increases during fasting or carbohydrate restriction, providing a vital alternative energy source for many peripheral tissues. In these extrahepatic tissues, like muscle, heart, kidney, and importantly the brain during adaptation, the process essentially runs in reverse, but with a key difference. First, if 3-hydroxybutyrate BHB is taken up, it's oxidized back to acetoacetate by the same enzyme, 3-hydroxybutyrate dehydrogenase, but this time the reaction produces NADH within the peripheral tissue's mitochondria, contributing to ATP production. Then comes the critical activation step for acetoacetate. Needs to be converted back into acetoacetyl-CoA to enter the final stage of breakdown. This is achieved by transferring a CoA molecule from 6-acyl-CoA, an intermediate of the TCA cycle, onto acetoacetate. This crucial reaction is catalyzed by an enzyme called 6-acyl-CoA.acetoacetate-CoA transferase, which is often referred to by its older name, the thiophorase.
Thiophorase transfers the CoA.
Yes. This enzyme effectively activates acetoacetate using a high-energy CoA donor readily available from the TCA cycle. While this thiophorase reaction is technically reversible, the product, acetoacetyl-CoA, is immediately cleaved by the thiolase enzyme, the same one used in beta-oxidation, into 2 molecules of acetyl-CoA. This rapid removal of the product pulls the thiophorase reaction strongly forward, ensuring efficient ketone body utilization. The resulting acetyl-CoA molecules can then directly enter the TCA cycle within that peripheral tissue to generate ATP.
So ketones get converted back to acetyl-CoA in peripheral tissues.
Exactly. Most extrahepatic tissues that have mitochondria, including the brain after adaptation, heart, skeletal muscle, kidney cortex, possess significant thiophorase activity and efficiently oxidize both acetoacetate and 3-hydroxybutyrate in this manner for energy. Notably, red blood cells lacking mitochondria cannot use ketones. And here's the key aha moment, the crucial piece of tissue specificity that often surprises people. The liver, despite being the primary site of massive ketone body production, almost completely lacks the thiophorase enzyme.
The liver can't activate the ketones it makes.
Precisely. The liver synthesizes ketone bodies specifically for export and use by other peripheral tissues, particularly during times of metabolic stress or glucose scarcity. It cannot significantly utilize them as fuel itself. It's a dedicated factory, generously producing energy currency for the rest of the body, especially the brain, but not using it locally.
That's a truly profound insight and a beautiful example of metabolic specialization. The liver makes them, but can't burn them itself. It's a selfless metabolic act, ensuring fuel availability for other critical organs. It's a remarkable division of labor.
It is indeed. And this specialization is absolutely essential for maintaining whole body energy balance, especially during prolonged fasting. However, this system can become overwhelmed. When the rate of ketone body formation in the liver significantly exceeds the rate at which peripheral tissues can effectively utilize them, their concentration begins to rise dramatically in the blood, a condition called ketonemia. As blood levels rise, ketones start to spill over into the urine, leading to ketonuria. If this process goes unchecked and ketone levels become excessively high, it can lead to a dangerous medical condition.
Ketoacidosis.
Exactly. Diabetic ketoacidosis, or DKA, is the most common and severe example. This typically occurs in individuals with uncontrolled type 1 diabetes mellitus, where there's an absolute deficiency of insulin coupled with high levels of counter-regulatory hormones like glucagon. In DKA, the lack of insulin and excess glucagon leads to unrestrained lipolysis in adipose tissue and unchecked fatty acid oxidation and ketogenesis in the liver. Blood ketone levels can soar to extremely high levels, sometimes exceeding 20-25 millimolar compared to normal levels, typically below 0.3 millimolar. Urinary excretion also becomes massive.
And these ketones are acids, right?
Yes. Acetoacetate and 3-hydroxybutyrate are organic acids. They have a carboxyl group with a pK around 4 to 5. At the physiological pH of blood, around 7.4, they readily dissociate, releasing a proton, H+, into the bloodstream. When ketone production is excessive, this massive release of protons overwhelms the body's buffering systems, like bicarbonate, causing the blood pH to drop significantly below the normal range, resulting in metabolic acidosis. Furthermore, in uncontrolled type 1 diabetes, the extremely high blood glucose levels also lead to osmotic diuresis, excessive urination, causing significant loss of water and electrolytes. So in DKA, you have this dangerous combination, an increased concentration of H plus from the ketone acid circulating in the significantly decreased volume of plasma water due to dehydration. This synergy leads to severe, life-threatening acidosis.
A perfect storm metabolically.
It truly is. A classic clinical symptom of DKA is that characteristic fruity or acetone-like odor on the patient's breath, which, as we discussed, is due to the increased spontaneous decarboxylation of acetoacetate to volatile acetone when levels are very high. While most commonly associated with uncontrolled type 1 diabetes, significant ketoacidosis can also, though usually less severely, occur in cases of prolonged starvation or severe fasting, and sometimes with excessive chronic ethanol consumption, which can also disrupt liver metabolism and promote ketogenesis.
This is where the intricacies of biochemistry directly translate into a potentially fatal medical emergency, powerfully highlighting the delicate metabolic balance your body constantly strives to maintain. It's not just abstract theory, it has very real, critical implications for health and survival.
Absolutely. Understanding these pathways is crucial for managing such conditions.
Wow. We've certainly journeyed through the incredibly intricate pathways of fatty acid synthesis, their breakdown via beta-oxidation, and the body's ingenious emergency fuel system of ketone bodies. From that initial pivotal spark of acetyl-CoA branching off in different directions, to the complex multi-layered regulation by hormones like insulin and glucagon, enzymes like ACC and HSL, and transporters like the carnitine shuttle, it's truly a symphony of metabolic reactions, orchestrated with astonishing precision and adaptability.
It really is. What's truly fascinating here, and what we hope you've gained a deeper appreciation for today, is the sheer elegance and, frankly, the brilliance of the body's energy management systems. Think about the reciprocal regulation of synthesis and degradation, how your body cleverly avoids making and breaking down fat simultaneously. Consider the specialized tissue-specific roles, like the liver's unique ability to make ketone bodies but not use them, acting as a dedicated fuel provider for others. And recall the crucial, non-negotiable role of essential fatty acids that must come from your diet. It all profoundly underscores the intricate interconnectedness and efficiency of metabolism. It is, without exaggeration, a masterclass in biochemical engineering refined over millennia for survival and optimal function.
So what does this all mean for you, listening right now? It means that every bite you take, every meal you skip, every period of fasting you might undertake, triggers a cascading sequence of these sophisticated biochemical events, all meticulously designed to keep you optimally fueled, your cell structures intact and functional, and your brain sharp and fully operational under varying conditions. The mastery you've hopefully gained today isn't just about, memorizing complex pathways or enzyme names. It's about genuinely appreciating the profound adaptability, the resilience, and the inherent intelligence of your own incredible biology working constantly behind the scenes.
Absolutely. And perhaps this deep dive, this exploration of how exquisitely tuned our fat metabolism is for both storing energy during abundance and mobilizing it during scarcity, raises an important, maybe even provocative question for you to ponder in our modern world. Given this incredible efficiency and the ancient, finely tuned adaptability of fat metabolism, designed over eons of intermittent food availability, what are the broader implications, the potential mismatches created by modern dietary patterns that so often provide a continuous, relentless, unchallenged excess of calories, potentially overwhelming or dysregulating these very systems that were designed for a much different rhythm of feast and famine?
That's a powerful thought to leave with. Keep that question in mind as you go about your day. We encourage you to observe how these fundamental concepts we've discussed, energy storage, fuel mobilization, hormonal control, essential nutrients, resonate with the constant stream of information you encounter daily about diet, health, energy levels, and metabolic well-being. Thank you for diving deep with us today into the world of fats and ketones. Until next time, stay curious and keep exploring the incredible world within you.